Hi everyone, welcome to the unit on immune disorders and vaccinations. Throughout this unit, we're going to look at two different sides of the immune system. What happens when things go wrong with the immune system, which can lead to disease and disorders, and then how can we use the immune system to our own benefit through vaccinations. So through this first video, we're going to look at different classifications of immune disorders, and we're going to focus on immunodeficiencies and autoimmune disorders. So what exactly is an immune disorder? Immune disorders are just a classification of disease where your immune system is not working the way it's supposed to. And there are four main types of these types of immune disorders. One is if you're born with a weakened immune system. Those are called primary immunodeficiency diseases. Or you could get a disease that actually weakens your immune system sometime later in life. Those are acquired immune deficiency diseases. Third type is if your immune system turns against itself, so it starts attacking self as opposed to foreign antigen, which are the autoimmune diseases. And then the fourth is if you have an overactive immune system, so it starts responding things that you don't really want it to respond to. These are called hypersensitivities. You also know them as allergies. We're going to take a look at the different types of diseases as we go. So we're going to start with primary immunodeficiencies. Your primary immunodeficiencies are genetic disorders, so they're inherited from your parents, and what happens is there's a defect or a mutation in one of the genes that's necessary for development of the immune system. Depending on what gene is mutated, it really changes the phenotype or the type of disease you have, but overall, most primary immunodeficiencies cause some sort of chronic increased susceptibility to infection. There are over 150 known primary immunodeficiency disorders. They're all really pretty rare, but most of them, so 50 to 60%, tend to be more in the B cells, whereas 5 to 10% are in the T cells, and the rest actually will happen in your innate immune cells. So for example, say you had a mutation in the gene here, leading for the development of a B lymphocyte to a plasma cell. Depending on what gene that was, it could be that you could make, say, IgG, but not IgM. Or maybe you could make most of your immunoglobulins. That's different than if you had a mutation in a gene up here for development where you don't make T cells or B cells, which means you have no adaptive immune system. That disease is called SCID. Or remember, we could have mutations in the development of your innate system. So for example, maybe you have a mutation here and you don't make any of the phagocytic cells. That would be a problem because you wouldn't have any antigen-presenting cells to activate the adaptive system. But if you had a mutation in the gene here, we would still make dendritic cells to activate the adaptive system, but we wouldn't have any macrophages to do phagocytosis. So this is why the phenotype, the actual disease, is dependent on what gene is actually part of the genetic disorder itself. Those SCID mutations, which stands for severe combined immunodeficiencies, are the most serious of the primary immunodeficiencies. These children are born without any adaptive immune system, so no B cells or T cells. And this picture here is actually David Vetter, who was known as the Bubble Boy. He was born in 1971, and he had SCID. He was born to parents who had actually had a child before him that died very early on because of SCID. Since it's a genetic disorder, they knew they might have another child that would have the same disorder. So when he was born, they were actually ready with a sterile chamber to put him in. And he lived for 12 years, but had to spend his entire time in this sterile bubble. The suit that you can see here in the picture was actually built by NASA, which was the first time he was able to come out of the bubble and actually walk around and hug his parents. Other than that, he had to be in a sterile environment because he had no immune system to deal with any possible foreign pathogens. I mean, his food had to be sterilized, his water had to be sterilized, and the air itself had to be sterilized. These days, SCID patients are treated with bone marrow transplants to try to give them a functioning immune system. Your primary immunodeficiencies are something you're born with. This is different than an acquired immunodeficiency, which is going to be something that you acquire at some point during your life. This can happen in a couple of ways. So one is that common infections can actually temporarily suppress the immune system. So these are acquired immunodeficiencies that are short-term. It's not something permanent that you have, but for a certain period of time, you're more susceptible to disease because your immune system is temporarily weaker. Some of the key ones, influenza and mononucleosis, which is better known as mono, both of those can temporarily suppress the immune system. And interestingly, measles infection will actually completely wipe out your memory cells that you would have already had against other diseases. So if you've been building memory cells to all these other pathogens and you get a measles infection, it wipes the slate clean and it makes you susceptible again to all those other pathogens you had already made memory cells for. The other way you can have acquired immunodeficiencies is that the immune cells themselves can be a target of an infection or a drug treatment. And so this leads to much more severe immune suppression and much more long-term immune suppression. So for example, HIV infection is going to infect your immune cells. That's causing those immune cells to die, which is leading to this acquired immunodeficiency. Another good example is people who have had organ transplants. They need to be on immunosuppressive drugs for the rest of their life. So they're going to have an acquired immunodeficiency because of the drugs they're taking to specifically tone down what their immune system can do. Interesting, while not considered immunodeficiency, cancer is a malfunction of the immune system. So cancer cells themselves are supposed to be recognized by your immune system as something that is no longer self. When you have cancer, it's because those cancerous cells escaped recognition and they were not destroyed by the immune system the way they're supposed to. And so really it's the cytotoxic T cells and your natural killer cells that should destroy these mutated cells before they can actually grow and proliferate into cancerous tumors. And we see that sometimes antigens do this better than other antigens. So for example, down here at the bottom are the normal cells. So you'd have a normal protein, remember, expressed out on MHC1. This is self, so nothing happens. But if you get a mutation in that protein, which happens in numerous genes in cancerous cells, now that protein looks different out on the surface, being expressed on the MHC1, so this should be recognized by those cytotoxic T cells. Other ones that are what we consider high tumor specificity, which means they usually are recognized, are if you have what's called an oncogenic virus. So some cancers are actually caused by a viral infection because the virus in the genome has genes that can make it cancerous. Things like cervical cancer is caused by the human papillomavirus infection. And so in that case, your normal cell doesn't have the gene because it doesn't have the virus infection. Your infected cell makes the protein. That protein is expressed and again recognized by the immune system. But then you have other cancers that are caused just because of a change in gene expression. So maybe this is a normal self protein that's supposed to be expressed, say, in the heart, where it's considered normal, but now it's being expressed in the liver. This might cause the liver to become cancerous, but it's a normal self protein, so your immune system doesn't really recognize it as well. Or similarly, you have a normal self protein that's expressed at a certain level. If you get overexpression of that, you're still just putting normal protein out. These are considered low specificity, and often the immune system doesn't recognize them the way they should. The next immune disorder we'll talk about are the autoimmune diseases. So autoimmune diseases are really when you get a breakdown in that self-tolerance. So this is when the immune system starts attacking your own tissues as opposed to something foreign. Okay? It's not supposed to do that. We're not supposed to recognize self-antigen. But sometimes when that happens, you can have autoantibodies, so antibodies that are binding to your own cells and tissues, or autoimmune T cells, T cells that are responding to your own cells or tissues. And if that happens, then your immune system is going to damage your own tissue the same way it would have damaged a foreign pathogen coming into the body. These autoimmune diseases can either be organ specific, so your immune system is only targeting and attacking one specific organ. We see this in type 1 diabetes. So type 1 diabetes is an autoimmune disease. It's when your cytotoxic T cells are actually attacking the islet cells of the pancreas, which are the ones that produce insulin. So this is why everyone who has type 1 diabetes needs to take insulin. It's because your immune system has actually destroyed all of the cells in the body that could produce insulin on their own. You can also have autoimmune diseases that are what we call systemic. They're attacking tissue throughout the entire body. A good example of this is lupus, which has autoantibodies that are binding lots of different antigens in all of your tissues. That gets stuck in your organs and ends up activating complement. Remember, that complement cascade can lyse your own cells just as easily as it can lyse bacterial cells. And so we start destroying cells among lots of tissues in our own body. Now, there's a couple of ways that autoimmune diseases can develop. The first, and probably one of the more common, is a defect in the negative selection during maturation processes. So remember, your lymphocytes were supposed to mature in either the bone marrow or the thymus, and as part of that process, step 2 was to remove any lymphocyte that was able to bind to self-antigen. If some of those slip through the cracks and end up in the secondary lymph node, so they actually mature and move on, then you have part of your pool of lymphocytes that's now able to bind to your own antigen, and that can cause problems in the body. The other way this can happen is if a microbial antigen looks a little bit like self, and so say it has one epitope that's clearly foreign, but a different epitope on the antigen is a self antigen. Sometimes when that antigen gets processed, and the body knows that it's foreign, but that self one also causes some activation, and we start producing antibodies similar to it, and those antibodies could then go off and cross-react, bind to our own self tissue as opposed to just the pathogen. So it was a pathogen protein, but because the proteins look similar, we're also able to then bind to our own cells and attack our own tissues. And then the third is that there are what we call hidden antigens. So things that are never leaving the cell will never actually be exposed to the immune system. Something like DNA. It's always in the nucleus. There's really no chance for that to be processed and put on the surface. There's no antibodies that are going to be recognizing it. But if there's some sort of trauma, so this happens sometimes after a viral infection that lyses cells, or if there's tissue damage, then those antigens that were hidden inside might actually be released into the body. Then they look foreign because now they're out of our cells where they weren't before, and the immune system responds to them as if they were foreign molecules. So those are 3 ideas of where these autoimmune diseases come from. But it's important for you guys to recognize that for the most part, we have a very low understanding of how autoimmune diseases actually develop. It's really kind of a big black box. There's a lot of research going into it right now, but we don't have a whole lot of details. And so you guys just need to understand the general overview of what could be causing these autoimmune diseases.